Everyone, guys, thank you for sticking around for a few more minutes. There's the talk title. So this actually was just recently published, so I'm pretty excited about that. So it's an article now that you can actually find this data published, so you can look it up for yourselves as well. But basically, Cell-Esson is a novel way to deliver lipids, fats as a supplement to bioproduction, in particular in CHO cells. And so what I'd like to do is actually just go through an overview of how proteins are made, because I think sometimes we just take for granted that you put into a bioproduction the CHO cells and out comes the antibodies. But basically everything happens on lipid membranes. So in the ER, you have a low cholesterol dense membrane. That's where the proteins are made. And then it goes out to the Golgi, where post-translational modification, in particular glycosylation, happens. And this is a high cholesterol dense membrane and requires that density in order to have good post-translational modifications, which affects your protein quality. And then you have it wrapped up into a membrane vesicle, which is secreted, and then the protein is then in the soup, where then it's purified. So basically, by adding Cell-Esson, you address the lipid stabilization and energetics component, which is a fairly important part. And so what we're going to show here is basically an open-ended experiment, but first let's just back up and talk about some of the components of Cell-Esson. So Cell-Esson is chemically defined, animal component free. It's manufactured under GMP guidelines, and what we found is actually you get reproducible behavior from lot to lot. And there's data that I'm not showing here, which is going to be coming up in posters, I guess, at Pep Talk would be the first time I'm going to present it. But basically there's lower variation in the glycosylation profile, and I'm going to show you the metabolic profile, but even between metabolites, there's less variation from run to run. So there's stability that happens from improved protein health. And it's unique mechanism which allows it to be used in single-use systems, which I'll show you at the tail end. So this is essentially broken up into three parts. The first part is proof of principle, where I'm showing a batch experiment. Second part is in a fed batch, and then I'm going to show you that it's scalable using the Amber model. And then the fourth part, still scalable, but then in a single-use system, showing that you can add lipids and cholesterol into a wave bag, which to date really hasn't been done successfully. And so these experiments were all done open-ended, where these lines over here show the potential outcomes. The green line shows that at any amount of Cell-Esson you add, you'll have a positive effect. The blue line shows that there's some optimal point where you'll get a benefit, and then after that there's no benefit. Red line shows that if you add Cell-Esson, there's going to be some detrimental effect. And then there's this flat line which shows that there will be no benefit by adding Cell-Esson. This is in a shake flask, and it was done in triplicate. But basically throughout, what you'll notice in this and in other models is that there's no effect on the proliferation. So essentially, Cell-Esson does not increase or decrease the proliferative capacity of the cell. But what it does do is increase the amount of titer that you get. And so when you look here, is that if you look at the percent of increase in bulk titer, is that there's a 20% increase in bulk titer at 1% of Cell-Esson when you add it at day 0 in a 7-day batch culture. But what you also notice is that there's a decrease in titer at 0.5% and 2.5%. So this resembles the blue line that I showed you, where there's an optimal point. And we found that this is also the case, is that there is an optimal point where Cell-Esson will have a benefit. Now, this is not just true for Cell-Esson, it also happens to be true for basic ingredients like glucose and glutamine. So essential ingredients that are added also have a strike point where they're no longer optimized. And so this is also true for Cell-Esson, is that there is a point where you'll find that optimal concentration. And so then if you look at this calculation, and you actually do the calculation around amount of protein made on a per cell basis, you'll see that there's an increase in productivity, which implies that there's a more efficient protein secretion, which goes back towards mechanism, which I suggested was improved membrane health. And then this was again validated if you look at it by QP, which is a more conservative way to measure productivity, where if you look at the integral under the curve at different amounts of Cell-Esson, that there's no big change in the amount of cells that are there. And this is, I showed you on the original proliferation curve. But then if you look on the amount of protein that's made across the time span, you'll see that there's actually an increased productivity measured by QP as well. And so that's, again, on a batch culture, that was proof of principle. But then if we move to using Cell-Esson as a feed in a fed batch culture, and again, same setup, open-ended experiment, and we fed it 3 different times. And again, what you'll see is that there's the same trend, and that is that you get an increase in titer. Here I'm just showing you an increase in titer over control, and this is being fed 3 times. And so the control basically is at 0, and here at 3 different amounts of Cell-Esson that's added as a feed, that you get a significant increase in protein production. And here at the tail end, that this was a 25% increase in titer. And so then, is it scalable? So far I've showed you I've showed you experiments that were done in shake flask, and I showed you that it was reproducible in a fed batch. And so then when you move to an amber system, and so in the amber model, this is in a amber 15, which they've shown is scalable up to, people have shown it up to 10,000, but definitely scalable up to 1,000 liters, that those results are scaled up. So essentially you see the same thing that I just showed you in a shake flask, which is that if you feed Cell-Esson, you get a significant increase in protein production, and again, this is in a 20% range. And what you'll also see is that the cell density is unchanged. So these data basically scaled from batch to the fed batch in shake flask, and now in the amber, the results are basically reproducible. The other thing that's interesting is that the first set of data I showed you was in an SAFC media, which is based in a GS0 cell line. This is in Lanza media, and this is a DG44. And so we've done this across multiple clones. And so the next set of data is I'm going to show you the metabolites from this. And essentially the metabolites are unchanged. And so they're all overlapping. So there's no change in the control at all from the control to Cell-Esson, which means that the increase in protein production doesn't affect any of the standard citric acid metabolites. So Cell-Esson is working via a different mechanism that's not within the TCA cycle. And I think that will go back, and I'll summarize that when we talk about mechanism, but none of the traditional things that you look at, glutamate, lactate, or ammonia, do you see any change in the metabolic features. And so the last part, which has been mentioned by several of the other speakers, is this movement towards using single-use systems. And in single-use systems, there's been a particular problem of adding any types of lipids, and in part because they adhere to the surface of the membrane. And so this is a study that was done at Merck, which basically demonstrates that after 72 hours, you lose the cholesterol that's solubilized in ethanol or cyclodextran. And these are the 2 commercially available options today. Neither one of these represent the Cell-Esson, but these are the 2 commercially available options. And in the standard single-use bags, which are either PA or EVA, after 72 hours, they're 90% gone. And so then if you look at linoleic acid, and they also measured linolenic acid, the majority of linolenic acid and linoleic acid are actually gone within 6 hours. So now, this is Cell-Esson in a single-use system. This is in a 40-CHO media. So now this is the 3rd type of media that I'm showing you. And basically what we have here is, again, showing you that there's no change in the proliferation. But interestingly, there's a benefit in viability. So the cells in the control started to basically die quickly, whereas in Cell-Esson, you see that there's a benefit where the cells continue to live. And so what we've seen is that in this tail end, the cells are actually the most productive. And so when you get increased viability at the tail end, you can actually get a lot more protein production. And this is especially true if you're looking at perfusion systems, where you want to basically have the cells continue on through 20 days. And here, if you have higher viabilities, you'll have much higher protein production. And so then if you actually look at titer, what you'll see at Titer is that if you compare the peak-to-peak production, this is a 27% increase in Titer. And if you look at the multiple days starting at the tail end, where in single-use systems you start to see the most production, that you get this increase in Titer that's significant. And so then, to summarize the talk, what I hope to have showed you is that you get increased Titer, and I've showed you that data, but we believe that this happens via two different mechanisms. One, increased stability of the membranes themselves, and so some of that data that I didn't show you here is that you get higher glycoforms when you use Cell-Esson, so you get more the G2F and more of the galactosylation, which shows that the Golgi apparatus is actually in better shape. The other component we think is that there's actually better energetics. So all of the lipid components and most of the media systems have to be made de novo. If you can supply those raw ingredients, you improve the energetics of the system. And so we believe that Cell-Esson basically improves both the energetics and the stability of the system, and that's how you get more protein secretion. And so what I showed you is that you can get greater than 30% protein increase, you get greater batch consistency, and that you can have large scale production from Cell-Esson. We make this under GMP.